Again, thank you for the opportunity to share my work and the work of my team on such an important day for Colby. All right, so let's see. Okay, so I'll start with thanking the team first up. Uh, I've been truly blessed to have these wonderful students working in my lab. It's a pleasure to work with them every day, and the kind of accomplishments that they have done for our work is just truly amazing. Let me highlight some of them. So this is the current lab right now, and these are all the alumni that have sort of graduated over the years. I'm going to continue to linger on this slide for a while so that you can recognize some of them, which are in your classes or in your performances or teams. Funding, of course, is helpful. So our lab has been blessed. We're launching a campaign today. NIH funding has really helped to run the lab, as well as the science division grant from Colby. So the work that I'll present today, I'll try to kind of create a story around it, was primarily done by these students, and then we are continuing on some of the work in the current lab. I would like to start with sort of talking a little bit more about this lab member who has been with me ever since, and we are currently working with it, and hopefully for many more years to come. So we work with fruit flies. All right, so it's a model organism to work with. Everything that we can do, fruit flies can do as well. They can fly and we cannot. So there's that. So they are capable of complex neurological behaviors, which you usually don't associate with them. They are perfectly fine for learning and memory type of work. And almost all major human behavior, like normal behavior and diseases, have been modeled in flies, and they're excellent in sort of reproducing some of the things that we feel, we respond, and they do the same way. So it creates a really nice model to work with them. And relevant for today's talk is this, the last point over here is we have this ability to take any human protein, mutant or normal, and introduce it inside the flies and ask what happens. Not only that, we can introduce this protein in the tissue that we want at the time we want. So this gives us this complete control over trying to understand function of a protein, take a human protein, bring it in flies, and ask what happens. And then what I'll try and do today is sort of talk a little bit about that kind of work, and as Margaret sort of mentioned, at a behavioral level, which is the most global of all, then at a cellular level, and then at a molecular level, and try and see if we can fix some of the damages that we're going to create. So flies are an excellent model to create diseases and understand them, and the disease that we're interested is in frontotemporal dementia. As the name suggests, it's the degeneration of frontal and temporal lobes. It's the second most prevalent disorder in dementia category after Alzheimer's disease, so huge biomedical implications. And the symptoms, if you're interested, include depression, apathy, lack of social tact, compulsive behavior. So it's a whole spectrum of things that are not functioning normally with the subjects. So there are genetic components that control some of this disorder. One mutation that was identified is in a gene called CHIMP2B. So anytime I sort of mention CHIMP2B, I'm talking about the gene and the protein that is related to frontotemporal dementia, or FTD. So here's a very sort of a simple schematic. A normal gene will make a protein which will be of, let's say, this length. Subjects who have the mutation will make a truncated protein. So anytime I will say CHIMP2B mutation, I'm talking about this truncated protein. So normal individuals will have a certain length of this protein, and then the subjects or the diseased individuals will have a shortened version. So as I said, fruit flies, we have the ability to take this protein, it's a human mutant protein associated with the disorder, put it in flies, and ask what happens. Okay? To start with, what we're going to do is we can put this protein anywhere in the fly body. As I said, we started doing that. The flies died, which was not good for us and them because we could not study a dead fly, right? So we started sort of expressing it only in the eyes. And again, later, if you have any questions, I can sort of talk about why we did that. So we are taking this human mutant protein and putting it in fly eyes and asking what happens. So this is what a fly eye looks like. Close-up of it. So here's the head. These are the two beautiful eyes. And here's a close-up of one of those eyes. And I can look at it all day, but so you can. Notice the symmetry in the structure. It's really sort of nice and precise. It's completely regulated. It's always the same size. These are compound eyes which are made up of 800 units. Every fly in each eye will have 800 units. So a fly has spent a lot of time and effort to make sure that every eye is composed up of 800 units. Okay? So let's see what happens when we put the mutant protein in. So we are putting chimp2b mutant only in fly eyes and asking how do they look like. That's how they look like. So you can, of course, easily see that the structure is somewhat disrupted. They're bulging, they're valleys. The precise symmetry is gone. You have more cells at places where you should not have. You have less places at other places. So you can easily tell the structure or the form is completely disrupted. Okay? So that's the sort of a morphology level. So the next obvious question is, well, how do these flies act? Are they performing the function that they're expecting of their eyes? Well, like humans, flies also use their eyes to see. So we need to give these flies an eye test, right? So sort of a drawback is that the flies will not be able to read, some sort of letters at a distance, or even if they are reading, we will not be able to understand if they're reading it or not, right? So we have to improvise a little bit here. So let's give flies an eye test. Okay? So here's a tube which is completely darkened. So the only thing is that this part is not blackened out. You put the flies in here, and then you ask, if you shine light on the other end, how many flies are going towards the light? So again, since we are launching a campaign today, I should mention, this is an extremely expensive piece of equipment. It is very expensive. It's a plastic tube with an electrical tape on it, right? So very expensive. All right, so wild-type flies, normal flies, overwhelming majority of the time will go towards the light. And we can ask them repeatedly and ask them how many times they're going to go towards the light. And as you can tell, they do that very consistently. So now, chimp2b mutant flies, mutation in the eyes, disrupted eye structure, they cannot really see the light. So they linger on the dark side a lot more, as you can tell. Only 40% of the flies can go towards the light. So what we have done is, we have not only disrupted the structure, we have also disrupted the function. So the next question becomes, what on earth this mutant protein is doing to these eyes to cause this deficit? It must be hijacking some sort of a common normal proteins inside the fruit fly eyes to do this. And that's the mechanism. If you can understand what proteins are misregulated, then we can begin to sort of address, can we rescue the defect? Okay? So for that, what the simplest way to do this is, what you do is you ask for, has anybody else seen the similar defect? And luckily for us, somebody had. So here's a fly which looks very similar to our chimp2b mutant fly. It also has the bulges in the eye. I don't know if it's projecting well or not, but like you can notice that they're like bulging in the eyes and misshapen structure. These flies, even though show the similar defect, have a completely unrelated sort of a thing with them. What they have is, they have too much of another protein called notch. So I've introduced you another protein here, notch. It's just a name of a protein, and if you have too much of it in the eyes look like this. So our next simplest question was, does chimp2b, the mutant form, causes eye defects by increasing notch levels? Right? And if that is true, all we have to do is reduce notch levels in these mutant flies and see if the eyes come back to normal shape. So we did that. You can, as I said, introduce a mutant protein or a human protein inside fly eyes. You can also take out their normal proteins away as well. So you see, they're really an awesome model. Okay, so again, I've shown you this before, the morphology, and I'm adding a little bit more detail to it a little bit. Now what you can do is you can just section these fly eyes and see what the internal structure looks like. So again, here's the wild-type fly eye. If you cut a section and if you're looking at a cross-section of it, what you'll notice is that, again, they have a really nice symmetry. And then if you begin to stain cells inside it, you'll see this really nice and pretty pattern, which is very sort of organized. In my chimp2b mutant flies, again, as you can see, the bulging, you can see much more nicely in the cross-section. This nice symmetry is gone. And notice how the layering of the cells or the patterning of the cells has been disrupted. So the structural deficits also go all the way inside the fly eyes. So these are the flies which are CHIM2B mutant with normal notch. Now what happens in the same mutant flies if I take the notch protein out or reduce it? And as you can see, there's almost a complete rescue of structure. So the flies have a much nicer shape, much more symmetrical. You can cut the section, and again, you can see the symmetry is reduced, and notice the improvement in the patterning. So does the function also improve? So we give these flies another eye test, and it has. So not only we have identified a defect, we have understood what the defect is for, or what is causing the defect, and then we were able to make the adjustment to rescue the defect. So pretty cool, isn't it, right? So we are able to sort of complete the whole story here. We can create a model, we can understand what's wrong in that model, and try and rescue it. Okay? So what are we up to lately? So here's another thing that is very common with neurodegenerative disorders is disruption of circadian rhythm. Most of the time, if you do a word association and you ask somebody circadian rhythm, the next answer is something related with sleep. But it does a lot more than sleep. It pretty much controls everything. It controls, of course, our nervous system, our sleep, reproduction, secretion, digestive system, hormonal release, as well as immunity. So it is pretty much controlling every physiological function of our body. Well, it's almost 2 o'clock, so we are somewhere here. Best coordination, fastest reaction time, right? It's going to go fairly quickly downhill, and that's when the sugar rush will come in, right? And then you need a 4 o'clock sugar rush, and you just keep doing this again and again. It's such an important function that everybody has to deal with it on a daily basis. So much so that this year the Nobel Committee recognized the description or the explanation for circadian rhythm by giving them the Nobel Prize this year. It happened like 4 weeks ago. Actually, they used the fruit flies as a model to describe that. And when I say they, it has a huge Maine connection to it. One of the recipients lives in Maine, retired from UMaine Orono. Another one is a Colby parent. So there's that. All right, so does CHIM2B mutant causes defects in circadian rhythm now? Right, so we are going a little bit deeper inside the brain now. Before we were only on the eyes, now we're asking what are the other deficits. So well, the question is, how can you measure circadian rhythm or sleep in flies? Even if they're keeping a sleep journal, it will be difficult for us to figure out what that sleep journal is. Right? So we have to, again, improvise a little bit that how are flies sleeping, how are flies not sleeping, that sort of a thing. So here's the improvisation. You put a fly in a tube, okay? And the proxy is if the fly is walking around, that means it's awake. If the fly is not walking around, that means it's asleep. So how do we know the fly is walking around or not? You put an infrared emitter and a detector. Flies cannot see this light, so they're not distracted that, hey, what's this shiny thing in the middle? Anytime they're walking, which they like to do anyway, they are breaking this beam. And the computer will count it as every time you break the beam, the computer will count those. All you do is you put these flies for a week inside these monitors, so that's an actual picture of the monitor. And you ask, what's the activity pattern over multiple days? That's the pattern. So flies on any given day will start, let's say, at 6 a.m. They do their job, right? They'll have an afternoon nap or sorts, and then they will active again in the evening, and then go to bed at 6 p.m. Right? They'll stay inactive, and then the next day, at the same time, they'll start do the same thing. And they'll keep doing this again and again every day at the same time. So that's the beauty of it, right? It's how reproducible it is, how consistent it is, and that's the normal 24-hour rhythm. So now you're wondering what happens to the CHIM2B mutant flies. This is what happens to them. So every day, once you start putting them in the monitor, they'll start somewhat normally. Notice that they're a little bit more noisy as well. They kind of stop a little bit early as well. But notice the major difference. Every day, they are waking up slightly earlier and earlier every day. So there is a behavioral deficit. There is some sort of a sleep disorder. There's some sort of an activity pattern disorder. Now the question is, what's the mechanism for that? Much like how we solved the story before with the eye deficits, now what we are working on is, well, what's really causing this? What's the cellular and molecular basis for this deficit? So in one really long sentence, what I've shown you today is expression of human mutant CHIM2B, which is associated with frontotemporal dementia, causes structural and functional deficits in fly eyes, which can be rescued by reducing notch levels. And also, this mutant causes circadian rhythm deficits, and for which we are currently determining the mechanism. And when I say we, that means they. Thank you. I'll be happy to answer any questions.
Thank you, Tariq. So, questions for Tariq? Yeah. Robert.
Thank you. That was terrific. I have a question about your NIH funding. How long is it, and have you noticed a change or risk to the funding relative to any electoral outcomes?
Okay, so that sort of touches various points. Yes, there are some. So it was for 5 years. It's still going on, and it will last until 2019. We are up for renewal. So all of Maine, so it's a Maine NIH grant. So multiple institutions in Maine will work collaboratively to apply for the renewal. There are some concerns about the next round of funding, how much money will be made available. There are some talks of even canceling the whole program. But there were a lot of petitions sent through the Congress to make it happen again, like not to cancel it, of course. There were some talks of reducing the funding, which happened actually, and then they were able to petition again, and they were able to restore some of that money back. But our goal is that at least the main INBRI was very successful. We had really good benchmarks in terms of all sorts of assessment that they do, and we are very confident that it will be renewed. It's an excellent opportunity, especially for a junior faculty. It just sets them up so well. It's very good funding.
Martha.
So I'm not sure if I missed this, but where do you introduce the CHIM2B when you're studying circadian rhythm? Is it also in the eye, or is there another part of the fly body?
So it's a combination of both eyes and certain parts of the brain. It might appear that we would think, in humans it happens that we use eyes to sense light, and that controls our ability to sleep and wake up. In fruit flies, they actually don't really use eyes primarily for circadian rhythm. There are other light sensors on their head. So if you take the eyes out completely, they would still be able to maintain some of the circadian rhythm. So you have to express some of this mutant protein in the circadian neurons that are controlling the circadian rhythm inside the fly brain. So it's a combination of both eyes and inside the brain, yes.
Okay, I want to take the moderator's presumption here. Aren't you all wondering, how do you get the little thing? Like, they're really little. How do you do this, right? What's the...
Well,
right.
That's why I have so many people, right? And I'm not doing that, right? So, no, and we have been talking about this a little bit. So the way I pitch it to students, it's not that hard for me to recruit students. All I tell them is it's about sleep and waking up. We also do a little bit of alcohol-related work, which I have not presented today. So I say circadian rhythm and alcohol. So a lot of students are interested as it is. And then what I do is I sort of tell them that it's very therapeutic and meditative in a way when you're sitting looking through these flies under a microscope and just taking literally the brains out. So it's a skill. It takes a little bit of training, but it happens within a day. A person should be able to at least learn how to chop heads off, and then another day how to get the brains out. So it sounds gory, but it's very sort of nice and interesting.
Any other questions?
And if anybody is interested, they can come to my lab and we'll show you firsthand how to do this, right? It will not take you long to learn how to do it.
Any other questions? Thank you, Tariq.
Thank you.